Hi, all. Welcome to Microbiology Lecture Series. And today we're going to talk about the early microbiologists, which we just labeled them early microbiologists. They didn't think of themselves as microbiologists. Most of them were maybe not even scientists. Maybe they were just inventors or labeled themselves kind of an engineer or something like that, or maybe they're just a general scientist and not an actual biologist. But their inventions or their research paved the way for microbiology. Okay, and so let's dive into that. The first one I kind of want to talk about is Robert Hooke. Robert Hooke, for many of you, may know him from like your general biology course because he's the individual that coined the term cell. And he coined the term cell because he was the first person, at least documented person, to examine a living cell under a microscope. So he created a microscope, and rudimentary microscope, maybe even our magnifying glasses that we have today are as good as his microscope during the time. But it was good enough that he could see cork cells, and cork cells being the inner layer underneath the bark of a tree, and they happened to be rectangular, and it reminded him of the rooms that monks used to live in, and they were called cells, and that's where the name came from, at least based on the writings and the documents of the time. That's where he got the name from. A couple other things about Hooke that might be of interest is that he may have been the first to describe gravity, and Newton may have borrowed the idea, for a lack of better term, borrowed the idea from Hooke. Newton and Hooke didn't like each other at all, and so, but based on writings that have been put forth, it looks to be like Hooke actually wrote about it first. Not to take anything from away from Newton, because he did the mathematics and stuff like that, but probably got the idea from Hooke, is what most evidence suggests. The reason why I talk about him from a standpoint of early microbiologists is because he kind of paved the way for other individuals to start designing better microscopes and examining cells and examining life, microscopic life. And the next one I want to talk about is Antony von Leeuwenhoek, and he is known as the father of microbiology, and he wasn't a scientist. He would probably consider himself an inventor, and he was very good at making microscopes, and he made all types of varieties of microscopes. He was probably, like, microscope wise, he was very good at bending glass or making the glass for the microscopes, and that's what gave him an advantage over Hooke and a lot of the other individuals that were scientists at the time. He was good at engineering microscopes. When he made good quality microscopes, he used them, and so he's credited with the first person to ever see sperm, the first person to see blood cells. I mean, he literally, if you could throw it on a slide, he looked at it and tried to describe it and talk about it, and that's why he's often considered the father of microbiology. Okay. Let's see. Francisco Redi may have been the first to do microbe research, I guess. Maybe not, but at least his research really kind of paved the way for other scientists to start doing more research. And so in the time of Francisco Redi, his work during this time, the idea with where life came from was spontaneous generation. Okay? So this was the idea that was accepted by the majority of the public, is that if you put out a piece of meat on a table and maggots form on it in a few days, that it was spontaneous generation, that higher being poofed it into existence. Well, Francisco Redi, he didn't believe that, and he designed an experiment to show that flies do not spontaneously generate. And so his experiment was fairly simple but powerful enough to show that indeed, if you put meat inside of a flask and cap it off, that it doesn't spoil with maggots. And if you put like a cloth over the top, the flies will actually lay their eggs on the top of the cloth because they're attracted to the smell. And the maggots might even hatch, but they can't enter and reach the meat. And this is how he really determined that, again, it's spontaneous generation. And then if you as a control, leave it open, and sure enough, maggots can get in there. So this was great. It was great research, and you would think, well, okay, in the late 1600s, then that should have been it. That should have been the last time we really hear about spontaneous generation, and that's why people get sick, and that's why, babies are made, and like everything. Like that's why bread molds. That's why lots of things happen. Okay, that's why mosquito larvae grow in ponds and things like this. It is because people, during the time, if people didn't see the actual process of the laying of the eggs or the mold spores landing on the bread, then they believed it was spontaneous generation. And unfortunately for human society, is his experiments really didn't do what he thought, or what maybe other scientists thought at the time it would do. It took over 200 years after his experiments to provide enough evidence to kind of refute the spontaneous generation claims by the general public. And really, I think the work that was done by Louis Pasteur was kind of that driving force. So often Pasteur's work is kind of credited with driving the Golden Age of Microbiology, or driving research in microbiology, and specifically in disease and disease outbreak. So Pasteur's work, if I think most people are somewhat familiar with it, not just his work on spontaneous generation and refuting spontaneous generation, but his work with pasteurization. He also devised a rabies vaccine. He generated, this glassware, which, is named after him, Pasteur glassware. He did all kinds of experiments, but one of the most famous experiments that he did was to show that indeed, if you take nutrient broth that we take and utilize constantly in microbiology labs, if you take a fluid that's got nutrients in it and you don't allow air to get and be exposed to that nutrient broth, then you can wait years and nothing will form. It'll still be sterile. There will be no microbes forming in that nutrient broth. But the second, literally, or hours later, but really we know, because we know now, that it's basically the second you expose that air to those nutrients, those microbes start generating. At the time, for him, because he didn't have the microscopes or the technology that we have today, for him it was hours or days, and then he would see this. And sometimes it was, these tails, he would do it like this. Sometimes he would just break the glass off. There's lots of ways in which he did experiments, and he did for a very, very long time. And really, we don't talk about this much, but Louis Pasteur's work on rebuttal of spontaneous generation, or showing that living organisms are not spontaneously generated, provided Charles Darwin with a massive amount of evidence. And these two shared research. We're not sure if they actually talked to each other. They went to similar meetings, medical meetings as each other, but there's not really documents of whether or not they sat next to each other and had a conversation. But they definitely read each other's work, and it shows up in Darwin's research on the evolution of by means of natural selection, that indeed the explanation for life is not spontaneous, that life's all around, it's in the air, it's in the water, it's all over the place, and it really gave Darwin a lot of evidence that he could go with. Okay, so from there, from about the 1860s on, spontaneous generation was less accepted as kind of an explanation for how people got sick or how things rot or spoil or whatnot, and it really started a huge wave of medical research on microbiology. Okay, so when that was really disproved, okay, we then often say it's the golden age of microbiology. That's when we start seeing things like Pasteur's hypothesis on how disease is caused by microorganisms, which we now consider the germ theory of disease. It's because of this era, this time period, where Pasteur puts this hypothesis out there, and lots of scientists grab a hold of it and go like, all right, we're testing it, boom, 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 boom. And, that's how the scientific process works, is you take that hypothesis and you test it over and over and over again, and if no one is refuting your hypothesis and they're supporting it, then we can elevate it, and it becomes elevated to a theory, and now we call that the germ theory of disease. From there, we start to see, like the early 1900s, we start to see, like in the United States, health departments popping up. We start to see, instead of just filtering water, we start to see the use of chemicals in water, whether that be good or not, but it helps with maintaining clean water sources. And then yes, the pandemic, the influenza pandemic of 1918 was a huge deal. It killed lots of people. But instead of saying spontaneous generation, or instead of saying we don't know what caused it and we're not going to dive into what caused it, people started researching it, and they started finding out, we're spreading this. It's us, it's humans, and the kid is sick and you're taking care of them, and then you spread it to your other children. And we really start this wave where now it becomes important to start looking at microbiology and disease and the relationship of microscopic organisms to human health, and we start developing antibiotics. First antibiotic, penicillin, in the early 1940s. Okay, it's hard to believe that right around World War II, the end of World War II, was when the first antibiotic was ever generated. Before that point, if there was no antibiotics, if you were sick, it was bed rest. Okay, and then vaccination and on and on and on, and this really just that big push in microbiology, and it's continued. I mean, we still do it today, and there's lots of research in microbiology currently going on all over the world right now. Okay, and part of that is because of outbreaks. We still have lots of diseases, so whether there's still people in the world that believe in spontaneous generation, there sure is, but now that's not an explanation. Now we try to find out why. Why is that happening? What's the causative agent? Okay, so what's causing the different flu variations? How are they mutating? All of this information, so we're better at finding out the causative agent, tracking it, and hopefully we get better at preventing it. And so the one that's probably on most people's mind is the one that we just went through, and that's the COVID outbreak, COVID-19 outbreak. And just to throw it out there that the world is so connected now that, in 1918 it took months and, even years for that influenza strand to get to different countries and cause people to become sick. In the matter of 3 months, okay, from January 20th to March 20th, we went from 4 countries that had COVID-19 to 165 countries that had COVID-19. So because the world's connected, these outbreaks become much quicker, which means the science has to be faster and better. And that's really kind of the role of microbiology in the world today. So next subject, we're going to kind of talk about the members of the microbial world, and we'll come back and discuss what kind of belongs in that field of study next time.